Okay, just a reminder to the speakers, this talks are being recorded and live streamed to YouTube.
Where do I get a link for that?
Yes.
I think we cannot use a link because apart from the acceptance, I didn't get any email from the team. So.
You mean the link for the YouTube? Is that what you?
Yeah.
Okay. Let me see.
I think I found it.
You did?
Yeah, if you go, there's a live on YouTube thing, and then if you click the button, like the menu down, it says copy streaming link.
Yeah. I don't want to give you the wrong link. I want to make sure I give you the right link.
Okay, I think let's start because this is quite a long session. Normally after 5 or 10 minutes, the number of participants double more or less. But yes, welcome to the very last session of the Biophysics in Africa Zoom conference. It's my pleasure to chair this last session, which will be on biophotonics. Biophotonics is in principle the use of light as a tool to investigate biological systems. And I've been enjoying this conference very much, and I'm sure that you will also enjoy this session, because looking at the titles and the abstracts, I'm sure these talks will be of the same outstanding standard as the rest of this conference. So the first speaker for this session is Ruan Vuljun from the University of Stellenbosch University, and he's going to talk about investigating single beam cars for microscopy applications. Right, Ruan, you can share your screen and then the floor is yours.
Okay, is my screen visible?
Yes.
Okay, perfect. Okay, good, thank you for the introduction. So I will be, as Chad said, talking about single beam cars for microscopy. I've done this work under the supervision of Erich Rover and Peter Neetling. So let's get into it. So cars microscopy is an imaging modality that is used to study biological samples. And I have some examples just from the internet to quickly show you like what some of these things can look like. So like at the top left here, we have some nerves that are imaged with cars, and fluorescence. So the blue here is a nerve cell, whereas the green is the myelin sheath, I think, that is imaged with cars. So it's really also like a complementary technique that you can use in conjunction with other imaging systems. It's also quite typically used to image lipids or fat cells in biological samples, as we see here. And it's also, again, superimposed with a different technique so that you can get a nice, more complete image of your biological system. What's nice about cars is that it's tag-free, so you don't have to change the biology of your system in order to image anything. You just have your lasers fired onto your sample and it generates some light, and then you can measure it to image the system. So CARS is the acronym for Coherent Antistock-Raman Scattering, which is a third-order nonlinear process. And I'm just going to quickly run over what this means, just quickly diagrammatically. So typically you have a resonant signal and a non-resonant signal. And to generate a resonant signal, you need to have a probe field and a Stokes field, which together interfere to excite your molecule to an excited vibrational state. A third field, which we call a probe, then induces an anti-Stokes photon to be emitted, and this is what we measure. So if we know what the probe frequency is and we've measured this frequency, then we can figure out what the frequency is of the resonant transition. For a non-resonant CAR signal, the Stokes field and pump fields different does not match a transition, but you're still able to generate this anti-Stokes field. So this is the resonant CARS is what we want, as this builds a fingerprint of the molecules that you want to study, whereas the non-resonant gives you information that you do not want. So for real systems, you obviously have more vibrational transitions. So in order to determine what the spectrum of this is, you'll have to change the frequency of your Stokes field so that you can excite it to those different vibrational levels or resonant levels, and then probe it again to get your anti-Stokes fields out. So normally to do this, a typical setup will have about 2 lasers, one laser that can act as your pump and your probe, and a third that you can scan these different frequencies to build up a spectrum of your molecule. So what this usually does is it makes your system quite complicated, as these fields also need to interact with each other at the sample at precise geometry. So they need to come in incident on your sample at exact angles so that you can phase match these things, which is an important thing for nonlinear processes. On top of that, these systems are usually quite expensive, which brings us to why we would want to look at something like single beam CARS, because it can be cheaper if you can do this all with one laser, and one laser also means that your geometry and your setup is less complicated. So for single beam CARS, moving away from this classical picture, we want one laser beam that can cover a range of frequencies instead of looking at one at a single time. So if you were to have this, I have a simulated broadband spectrum here of about 100 nanometers. If this was your excitation spectrum for one beam, we can calculate what the, probability is of a particular transition to be excited. And this is what I have plotted here on the right. So this spectrum here can simultaneously excite a broad range of Raman frequencies instead of looking at them one by one and without tuning anything. So if you were to then again have this spectrum to interact with each other and act as the probe rather, you will generate a CAR signal that looks like this in the blue. So the first thing you notice is that this CAR signal is quite blue and it's devoid of any Raman resonances. It's only the smooth function here, which already tells you there's some work that needs to be done with a single beam CAR setup before you can get any sensible information out of it. So with this in mind, we have a research strategy, and which also will be my outline for this talk, as building the foundation of the single beam CAR setup. Starting there, and this setup needs to have a broadband light source. It needs to have phase control of the light source. So this will be important to get rid of that broad response that we saw in the previous slide. And it also needs to be able to fulfill phase matching conditions. Once this is all in place, you can move forward and think about single beam CAR strategies that can recover narrow resonances in your measurement and also target chosen resonances. So if we can have this targeting here in place, That means we can move forward to a microscopy system where we can isolate a particular transition that we know that's contained within a biological sample and only image that particular molecule in this 2D space. Okay, so what our setup looks like is represented in this diagram. And it starts off with a normal femtosecond oscillator with some laser outputs at the right, on the right, for those of you who are interested. We use this laser oscillator to pump a photonic crystal fiber, which produces a broadband excitation source that's needed. So the PCF, together with the laser source, takes care of our broadband light. The light that's produced by the PCF is taken to a pulse shaper, which is a grating pair and a lens pair in a 4F shaper geometry with a spatial light modulator placed at the center. So what's special about this is light that hits the grating is dispersed, or rather the light is dispersed into its frequency components, and this lens L2 does a one-to-one mapping of frequency component to spatial coordinates in this plane where the SLN is placed. So that means we have frequency components here occupying their own space. If we know where these particular frequency components are, then we can use a spatial light modulator to affect the phase and polarization of those components. If you can affect the phase of the thing, then you can shape your pulse arbitrarily how you want. This light propagates through and is focused with length L3 onto grating 2, which then bundles everything back together in a nice packet. The laser is then focused with a microscope objective onto a sample, and this microscope objective takes care of your phase matching. So all three very important things that were needed for the single beam car setup is taken care of in here with these three main steps. Sorry?
I'm sorry, go ahead.
Okay. A drawback of using a PCF to produce the broadband light source is that due to dispersion effects as a result of the material, the produced pulse is also stretched out in time. So the main function for this PCF is to take a bandwidth of light and increase it, but also as a result of the material, it gets stretched out in time. This result is not great for nonlinear processes, as you want something that is shorter in time as the same pulse that's shorter in time as a higher peak intensity. A higher peak intensity results in a more efficient nonlinear process. So without having, so you have to deal with this broad pulse before you can start with any single beam cars measurements. And to deal with this, our group came up with a reconstruction or a characterization method called I²π. And essentially what this does is it uses sequential measurements by taking your pulse, chirping it for different chirp values, so essentially stretching it out in time, and measuring a produced second harmonic. So if you concatenate all of these second harmonic spectra for different chirp values, you generate a spectrogram such as this. Running this through the algorithm, without getting into too much detail, can reconstruct the phase of our supercontinuum. If we know the supercontinuum's phase, then we can use the SLM again to make it flat, which is a flat phase then corresponds to a compressed pulse. So we also made so we tested this method by also comparing it to a different well-known and well-used or widely used technique called MIPS. And we took the supercontinuous spectrum, which looks like this, took a portion of it, compressed it using I²π and MIPS, and had it focused on a PBO to generate a second harmonic spectrum. As you can see, the measured second harmonic spectrum, which corresponds to MIPS, is much narrower and also lower intensities compared to that generated with I²π. So this confirms to us that our I²π works much better than MIPS, and is also a secondary, or rather an indirect way to show how well our pulse is compressed. We did the same to generate a normal single beam car spectrum with the 2 compressed pulses, and we again see an improvement. We also continue to do some other measurements, which you can look at in our published paper from last year, for those of you who are interested. Okay, so that means our setup is sorted, which means we can move on to looking at single beam car strategies which are able to recover narrow resonances and target chosen resonances. Okay, so for this we have to introduce another concept which is called spectral focusing. And what this is a technique that was implemented initially in classical cars, where you have 2 separate pump and Stokes beams. As you can see here at A, we have a pump beam and a Stokes beam in its time and frequency, and all of the frequency components overlap in time, which means you generate a broad excitation spectrum. So if you were to add 2 identical pieces of glass in the beam lines for these 2 beams, then you will chirp it linearly in time, and you will have only certain frequency components overlapping in time. And also these components are all a constant, let's say, spectral distance away from each other. And what if this effectively does is it focuses your entire spectral bandwidth to only target a much smaller region in your excitation, or rather in your chosen excitation spectrum. To tailor this to target different excitations, you can simply introduce a time delay in your pump or Stokes, which changes this spectral distance and then changes what frequency you target. So this is how it works classically, and we were able to port this to our single beam car setup by knowing that a linear chirp is in a frequency domain is a quadratic phase function. And we know that using an SLM, we can add phase functions onto our supercontinuum. So the first step is to take our excitation source and to chop it up into 3 regions: a probe, a Stokes, and a pump section. The probe is simply a narrow band that we've also changed the polarization to be orthogonal to the excitation. And in doing so, you recover narrow resonance structures in your spectrum again. For the remainder of the spectrum, the Stokes in the pump is simply shaped with a quadratic phase function. And what this essentially does is it separates your spectrum into spectral pairs that are always in phase. So you'll see this frequency component is always in phase with that one. This one is always in phase with that one, and out of phase with everything else around it. So that means with all of these frequency pairs that are in phase with each other, all at the same distance from each other, you'll have a preference for a specific transition that you are exciting. So if we were to calculate the population amplitude for something like this, you'll see the result here. And let's just focus for now on the green and the blue graphs. So for a specific steepness of this graph, you get a specific population amplitude. And as you see, from green to blue, we've increased the steepness of the quadratic phase function, and so we have narrowed the response around our chosen frequency. So we set the distance between the two quadratic phase functions to match 800 wave numbers, and we set the steepness of the quadratic phase function to narrow the possibility of exciting only around 800. So you see 800 as a nice peak and everything around it is suppressed, so which is good for targeting that specific transition. You can continue increasing this to make this response even narrower if you were to wanted to do that. So for those of you...
2 minutes left.
How many?
2 minutes.
2 minutes left. Oh, sorry.
Yes.
Okay, let me just skip that one. So we were able to do a measurement of this. So this is a a sample of paraxylene, and we were able to target particular Raman transitions while suppressing everything around it. So that's a success. And then I'm just going to skip to this last one. So as a proxy for a biological sample, we used olive oil and scanned it for different targeting, by targeting different Raman transitions, and we were able to identify well-known vibrational modes that you find in olive oil. So these modes here correspond to Raman transitions that are associated with fatty acids, which you will find in biological samples as well. So in doing so, we've confirmed that we should be able to, with our single beam CARS method or strategy, we should be able to isolate a vibrational mode, and in doing so, image particular biological samples. And with that, I say thank you.
Hey, thank you very much for a very interesting talk, Ruan.
Yeah, sorry, that went a bit longer.
No, it was perfect. You actually managed to wrap up your talk very well. Yeah, so single beam CARS is actually a very sophisticated technique. Actually, it's a set of techniques, and some of them being home developed. So that's really nice to see. So the floor is now open for questions.
I have a quick question.
Okay.
Is it possible for you to do this in a time resolved way so that you can do a time sequence of?
Yes, you can. So by changing with the SLM, you're also able to shift the probe around, so you can probe it at different times.
Yep.
So it's definitely possible. I've done some measurements like that. But the drawback is that the SLM has a limit to how far in time you can shift the probe, and it's not a lot.
Okay, there's time for another question.
I was going to say this is a method that could be complementary to neutron studies. To like if you could track a key hydrogen bond in time, you can see sort of what is the fate of a proton in the active site of some enzyme or something. That's a CARS technique.
I wouldn't know.
Yeah.
Okay, any other question? Okay, if not, let's give Ruan another hand of applause. And if you have more questions, you are welcome to continue in the chat during the next talk. So thank you very much, Ruan.
Thank you.
For an excellent talk. Right, so the next speaker is George Dwapanyin, and he's also from the University of Stellenbosch, and he's going to talk more about I squared pi, which has been briefly introduced by Ruan, but George is going to present this more elaborately, and the title of his talk is Improved Quality in Nonlinear Optical Imaging Using I squared pi Pulse Compression. Right, George, you can share your screen, and then the floor is yours.
Okay. I think I just shared my screen 2 seconds ago, so why can't I share it now again? Can you see my screen now?
No, not yet.
Not yet. Do you see the green button at the bottom?
Yeah, click share screen. Like this.
So you should. Is it taking a while to come up?
What did I see? Not your screen.
There you go.
Yeah, okay. So let me just share this one now. So now it's okay. Let me just track this one. Yeah, share. Is it okay now? Can everyone see?
Yes, that looks good. So
All right, all right. So thank you for having me, and I'm presenting on the improved image quality, and thank you, Ruwan, for making my work a little bit easier. So Ruwan spoke on the application of this novel I2Pi technique in cars, but I'm also going to focus on show the other side of it in relation to nonlinear microscopy. So basically, when we say nonlinear microscopy, we're just talking about microscopy, which is based on nonlinear optics. So microscopy techniques that make an appearance with in the presence of high intensity laser beams interacting with matter. So basically, with the normal nonlinear microscopy, you have a laser source, preferably an ultra-fast one. You focus on a certain sample which you have in your image plane, and then the nonlinear interactions of the light and the matter generate a certain signal, which you use some detector, depending on what imaging technique you're using, you collect your signals. So nonlinear microscopy is preferred mostly because you have a higher spatial resolution, and the near infrared laser lens, near infrared wavelengths that are used also help in probing deeper into biological tissues. Now, the whole thing is, if you have this image and you want to get better, then you can either increase the energy that you are depositing, so you increase your power, and then for a fixed repetition rate laser and a fixed area of illumination, if you increase your power, you should get more signals. But then the downside of increasing the energy is that you now need the probe, and gives option for higher photo damage because you're depositing more energy in a small space. Then you have higher thermal effects and then photo bleaching. But then again, if you now decrease the pulse duration of your laser, then for even smaller pulse energies, you can have higher peak intensities. And it's these peak intensities that drive the nonlinear responses. Now, if we look at the uncertainty principle, so between the spectral bandwidth and the pulse duration, realize that if you have a broader spectral width, then there is the option, or you are now better off having a shorter pulse duration. So spectral broadening can be achieved with the super continuum generation, as briefly mentioned by Rwanda. So our aim was to now design a setup for nonlinear microscopy using super continuum generation, because we know that it gives us the range and then a uniform spatial profile that we can compress our pulses. And then once we have this super continuum, then we can generate shorter pulses, which will now lead to higher peak intensities at reduced excitation energies. And then we can have a reduced risk of damage. Then we also want to show this first time that we are applying this novel I2Pi technique in nonlinear microscopy. So in a sense, we developed a setup which is somehow similar to the one Ruwan talked about. But the setup we have is also in 3 phases. So we have a super continuum generation path, then where we do the pulse characterization and compression, and then where we have our imaging. Now, we would also want to know that, since I said initially in the opening slides that normal nonlinear microscopy, you just have a laser, you have some imaging optics, and then so to mimic that, we have a path here which doesn't go through the super continuum. It's just a normal femtosecond laser, and then we go into our imaging system. So we're just going to look at the various sector. So the super continuum generation, as previously also mentioned, is basically what we're doing is focusing light into a photonic crystal fiber. Now, depending on the core of the fiber, you can either have it non-polarization maintaining or polarization maintaining. So in a sense, you come in with, you pump the fiber with a chirp-free pulse. Now the intensities within the fiber for a photonic crystal fiber is predominantly dominated by surface modulation. So the surface modulation would lead to generation of newer frequencies. And then the material dispersion property of the material itself will also lead to the pulse broadening in time. So you have a pulse with new frequencies as well as chirped. So you have pulse broadened. Now, typically there are different types of fibers. Those that are pumped in the anomalous region, so they have a positive dispersion, and those pumped in the normal region with a negative dispersion. Now, if we have it, this here is a profile of the fiber that we used. So we're pumping at 800, then we are in the normal regime. And that is hence we call it the all-normal dispersion based on the wavelength that we are pumping. Now, the advantage of this normal dispersion is that if you look at the profile here, so you have a uniform spatial profile of a supercontinuum. So the higher the pumping energy, the broader your spectral width that you generate. But then in the time, you look at it in time, you have a one-to-one mapping of the spectral components in time. So you can now easily have a stable pulse, which you can now temporarily manipulate with a spatial light modulator. Unlike the anomalous, where you have, for instance, at this point, you have two different components. So it makes manipulating these pulses in time a bit difficult, and then also unreliable because of fluctuations. And this is predominantly due to soliton effects that predominate in the anomalous region. So if we basically have the pump, and then we pump our supercontinuum, our PCF, then we come in with a normal pulse, so around 800. We pump our fiber and we generate a supercontinuum. So this is typically of around a pumping input power of around 200 milliwatts. Then you generate, so you move from a 13 nanometer bandwidth to around 110 for that wavelength. And assuming with this broadened pulse and then taking into account the uncertainty principle, then assuming there are no dispersions within, you should be able to compress this pulse to get a pulse duration of around 11 femtoseconds from the initial one, which is around 70, which is coming out from the oscillator. So it shows that if you have a broader pulse, you can get a smaller pulse in time. So the different techniques that people have used in compressing pulses, some use just chirp mirrors, some use rotating cylinders, and some use the temporal pulse compression algorithm, which is what we also focus on. So we have seen how we generate the supercontinuum, and then how do we do a pulse characterization and compression? So we do have chirp mirrors, which take care of they reduce the adjacent pixels on the SLM so that it reduces the efficiency of the SLM, as well as reduce the second order dispersion. So it reduces it and basically takes a lot of pressure off the SLM. And like my colleague Ruan said, you have a one-to-one mapping of the spectral components onto the different pixels, so you can manipulate them easily. And to do this phase measurement, we use a second order, a nonlinear crystal. So in this case, a BBU, where we generate the second harmonics and collect with a spectrometer, which is connected to a computer. So it creates a feedback loop from the SLM and the system. So we can physically measure what the phase is, and then at the same time, iteratively add it onto the SLM to generate our compressed pulses. Now, MIPS is what a lot of people have been using, which was pioneered by the Dantus group in Michigan. So basically what they do is you measure the spectrum iteratively, and for each different spectrum measurements, you compare where you have your highest second harmonic, which corresponds to where you have zero phase difference. And if you have a spectrum with which is equidistant, parallel, then it gives an idea of how well you've compressed your pulse. And from that, you're able to retrieve your measured phase, which we have here over our supercontinuum plotted around the relative angular frequency here. Now, the advantage of MIPS, why people use it a lot, it's a single beam method. So you have everything in one path. You don't need multiple beams. You can actually do simultaneous, like I said, there's a feedback. So you can do simultaneous phase measurement and pulse compression. And it's insensitive to phase matching. So you don't really worry about that. And everything happens in the sample plane. So you can add different optics. And you know, when you add different optics to your beam path, the phase changes. But since everything is happening in the sample plane, you can have different optics in your sample plane, and you'll still be able to compress these pulse. The downside of it, however, is that it's limited to its effective is limited to second order, and you need repetitive measurements, and you can only reconstruct the phase. So in view of that, the I square pi technique. So basically, you also have a certain transfer function where you come in with an initial guess, and then you create an output field in time. Now you convolute this field with itself to generate an object field in time. And because you measure the square of the output field from here, that is why we have an I square. So basically intrinsic square. So the square of the intrinsic tychographic iterative engine, that's the full meaning of I square pi. And from that, because you have it in time, you do Fourier transform, you get a field also in frequency. Now you maintain the phase, take the amplitude, and because it's a square here, you take the square root of it to get the amplitude of the field. Then you have it, you can now do a Fourier transform of this exit field in frequency. And from there, you can update it into your typographic iterative engine, and then you generate your output field in time. And this new output field, you can now do a Fourier transform and have the measured phase. So basically what happens is you measure, so you take these spectra, and then you have a single beam. So unlike the MIPS where you have multiple, you need multiple spectra. I square, you just need one single spectrum. And then from there, you are able to reconstruct the phase that we have here. Now, if you compare the two, to the eye, you see that the I square pi looks a bit worse. But then if we now map what the MIPS gives and what the I square gives, here, if you overlay the two, you see that there's more features in the I square pi. The I square pi has more structure than the MIPS. This shows that it's more sensitive to the complex spectral profile, phase profile of the supercontinuums generated. Now, if it's more sensitive, then it means it gives greater accurate phase measurements. And because it's a single shot, it's faster, and it also has the ability to reconstruct both the phase and the amplitude. So aside all these added advantages, it has all the advantages of MIPS as well. So we have that. And then we now add these phases. So these phases that you measure with the SLM, if you take the negative of these measured phases and then you add it onto the SLM, then you've created a near transform limited pulse, or you compress the pulse. So how do we do it? We now, to verify it, we measure the pulse durations. And here we use the non-interferometric collinear autocorrelation measurements. Now, like I said, some people can only do pulse compression just using chirped mirrors. So obviously the supercontinuum, as soon as it bounces off the chirped mirrors, we know that it's compressed, but to what extent? So we measured that one here. And then when we do the compression with MIPS, what is the pulse duration? We also measured here. And then we also use the I square pi technique here. And we see that if you look at the pulse duration that are measured with the I square pi, we have around 17 or around 18 femtoseconds. And then MIPS gives us around 21 femtoseconds. Just the chirp mirrors gives 20 femtoseconds, 25 femtoseconds, which shows that it's doing a lot in taking care of most of the higher or the second order dispersions. For the fundamental, we have it around 160 femtoseconds, which is basically what we measure after the beam traverses all the optics. So this is the pulse duration in the sample plane. So all these pulse durations are measured in the sample plane at the exact place where the sample is.
George, you have 2 minutes left.
All right. So with this, you are able to also see that the spatial profile doesn't change. We are able to measure the pulse width. So if we have the same pulse width, then we know that if the pulse width is the same and the repetition rate is the same, the only thing that will affect the peak intensity is the pulse duration, right? Which we have the beams here. So we applied it, measured. You see that with MIPS and I square pi, you have higher intensities here in BBU. Then again, we applied it in biological cells. So here we have pig skin, so the epidermal layer of pig skin. This is when we use the fundamental. This is when we just use Sharp mirror compress. This is when we use MIPS. This is when we use I square pi. And for each, we calculated the contrast and the signal to noise ratios. You see that with the I square pi, you have higher contrast, higher signal to noise ratios than MIPS. And then we compare the enhancement factors. You have about 2 times contrast, and then for MIPS, between MIPS and I square pi, and then about 2 times signal to noise enhancement. I think if you also look at it in cells, so these are mouse neuronal cells, 2 photon excitation. So we can do second harmonic, we can do 2 photon excitation. And again, you see that there's about twice signal improvement between the MIPS, which is common, and then the I square pi that we have. Again, the signal to noise we have, and we see greater details and more features in it. We can have more, read more about it in our paper in Joseph B. So we just know that from this, you see that the I square pi improves the accuracy of the measurement and compression. It's effective. So if you now have lesser powers, you can have less photo damage in tissues, and you can get more higher peak intensity, that's lower excitation energies. Greater contrast, greater signal to noise, meaning you have improved resolution, and the repetition rates of the laser makes it a potential application for faster scanning. So with that, thank you, and I'll be available to answer any questions that you have. Thank you.
Thank you very much, George, for a very nice talk. So it's really nice to see this home-developed technique and all the benefits that it offers, and you've also presented it very nicely, so thank you very much for that. I'm looking forward to see all the applications of this technique in the near future. Right, the floor is now open for questions. Okay, while the audience is thinking about questions, maybe a quick one from my side. So yeah, your very last point you've mentioned is that 80 megahertz operation has the potential for fast scanning. This is, well, the speed of scanning could be indirectly related to the resolution. So there should also be a limit in the in a rep rate that you can use. So what in your view would be the ultimate rep rate for nonlinear imaging, considering current technologies?
Well, like I said, these ones, in my imaging, for instance, I was doing point-by-point scanning. I wasn't using Galvo stages, which is more preferable in nonlinear micro. I was using Galvo scanners. So, I mean, if you have I think the higher the rep rate, the lesser the dwell time. So, and if you have higher energies, higher peak intensity, and you have less dwell time, you're still able to get it. It makes it better. But in terms of the actual, quantifying and putting an actual measure on what the ideal rep rate would be, I think I'll have to think about it further and get back to you. But I know that the higher the repetition rate, the lesser the dwell time, the lesser the damage. If you have higher peak intensity to drive your nonlinear signals. Yeah.
Okay, yeah, so yeah, the peak intensity, that's obviously the important measure in this case. Another question from the audience. It seems like you've overwhelmed the audience with the power of the technique.
Either overwhelmed or confused them. It's one of them.
Yeah, no, it's really a very nice technique. Thank you very much for your presentation, George. And yeah, once again, I would like to invite everyone, if you have more questions, please do not hesitate to use the chat function. Even if it's just a very basic question. I mean, the idea of this presentation, of this conference, is that we expose everyone to the nice work that's being done in Africa, and we would like everyone to learn something about it at whatever level it is. So thank you very much, George.
Thank you.
Right, so let's move on to our next speaker, who is Amadou Wagi, if I pronounced that correctly. And he's from he's affiliated with the African Physical Society, and will be talking about laser spectroscopy for plants monitoring and medical diagnostic and therapy. Right, Amadou, you can share your screen and then the floor is yours.
Okay. Do you hear me? Do you hear me?
Yes.
Very well. We see your slides.
Do you hear me?
Yes, we can hear you.
Okay. Thank you very much. I would like to first of all to thank the organizer for such a wonderful initiative, and especially I want to thank Lawrence Norris, who brings this Biophysics Day to the African Physical Society. Presently, I am the president of this African Physical Society. So I will start. I will make okay. I will. So the outline. I will make a brief presentation of the African Laser Atomic, Molecular, and Optical Science Network, and also a brief presentation of the African Physical Society. And in the second part, I will talk about the laser and LED. LED means light emitting diode, induced fluorescence spectroscopy in plant, and also laser induced breakdown spectroscopy in plant and in medical application. But for X-ray fluorescence spectroscopy, I will skip this part, but I will talk about the multispectral laser emitting diode microscopy. Okay, so for the network, no, it is important because all the work we have done concerning the laser spectroscopy in plant monitoring, in medicine, it was done in the framework of this African Laser Atomic, Molecular, and Optical Science Network, which was launched in Dakar in 1991 at the occasion of the first international workshop. So this is the main map of showing the different part where this network is represented. And I have to say that the network is also at the origin of the creation of the African Laser Center. I am an ex-officio member of African Laser Center, which was launched in Pretoria in 2003. And of course, who is speaking? Okay. So the different activity of the network is workshop, school, conference, development and cooperation, training program activities in international society, because the LAM is an international member, an international society member of optics, of the International Commission for Optics. And now for the presentation of the African Physical Society, it was under the leadership of Francis Alote, and the African physicists have been calling for the formation of a multinational professional society dedicated to African physicists. And in August 1983, number of African physicists and mathematicians convened to form the Society of African Physicists and Mathematicians, the SAPAM. It was the precursor of the African Physical Society, and it served as a primary professional organization for practicing physicists and mathematicians. And in November 2007, at a summit in Cape Town, I was there, in South Africa, with numerous of physicists, a national society in Africa, together with also National Society of Black Physicists. The SAPAM formally announced that it could become the African Physical Society. And it was decided to convey the official launching of the society during the forthcoming meeting of the LAM network in Senegal. It was, I think it was the LAM 10 meeting in Senegal. So the African Physical Society held the launching official ceremony in January 12, 2010 in Senegal. And Francis Alote, who passed away in 2017, I want to pray for his soul, was the president of the African Physical Society. And the AFPS is incorporated in Ghana. And the main goal is to promote and future research in physics in Africa in order to enhance technological, economical, and social cultural development in Africa, promote effective contact and cooperation among African physicists, and to collaborate with other society and international organizations in promoting scientific activity in Africa. And the African Physical Society has to serve as invaluable resources to all African and help leverage their communication, research, and collaboration. And I have to say that African Physical Society was among the main organizer of the International Year of Light. At this time, Professor Alote, with president of the European Physical Society, and also with Ghana country, with Russia, with Saudi Arabia, they bring the project of the International Year of Light at the UNESCO, and after they bring it to the United Nations, who declare 2015 year as the International Year of Light. So, and also in 2018, we organized the international conference in Kigali to convey to the revival of the African Physical Society after the pass away of the first president. And last year, we organized an online Zoom international conference in collaboration with ICTP and African Light Source. Now, I have to say that presently, the African Physical Society is mainly supported by ICTP. And also, we are among the main organizer of this meeting, Biophysics in Africa. Okay, now, this is just to show you some photo for LAM meeting, tropical meeting in Senegal. And this is the launching of the African Physical Society during the LAM 9 meeting in Senegal. Here you can see the president of Senegal. It was the reception at the presidential palace, and there is some leading people of the African Physical Society. Here you can see Professor Alote, you can see Sekazi Mitinga, you can see myself, Magruder, Kedro from Ivory Coast, from Morocco, Anthony from US, from South Africa, and many other people are here from Morocco and so on for the meeting, the launching meeting of the African Physical Society. And here we have the council which have been formed in Senegal. But in Kigali also we have now a new council with all representatives from all the parts of Africa. Here you can see people. Alote is sitting here. Myself. This is Najet from Tunisia. Kedro. No, this is Kwato from Cameroon, and also we have international people. Maza from South Africa. Adedoyin, who passed away a few time ago. I pray also for him. Joe Nimela, and you see, and also Singo, who was the president of the African, the chairman of the African Laser Center in the past, so and so on. And here in 2014 we create also the African Optic and Photonic Society with the representative of all the international societies. This is Sun Swanberg from Sweden, Anthony Johnson from Optical Society of America, Alote, myself, the dean, the Yatagai, the representative of SPI, and so on. There was all the optical societies there for this meeting. And this is some group photo of the LAM 10 conference for the launching of African Optic and Photonic Society. And this is for the International Year of Light. Also, African Physical Society have organized in Ghana in September a big meeting. This was a meeting that I organized in Senegal during for the celebration of the International Year of Light. But now let us start my talk with laser-induced fluorescence spectroscopy. the laser-induced fluorescence spectroscopy technique is used widely in research for a variety of applications, from interrogation of plasma plume to determine the determination of cancer cell, fluorescence spectroscopy of single molecule. And it is one of the most sensitive approach available for analytical purpose. It is easy to implement, well investigated, largely non-invasive, and imperative for many applications in environmental monitoring. Here, this is the principle of fluorescence spectroscopy with a Jablonski diagram. You see, you have the excitation, we have the relaxation processes, and you have the fluorescence emission and so on. This is, and it could be at room temperature. You have the absorption by molecule to produce either the first S1 or second S2 excited state, and after this excited state are decaying by fluorescence or phosphorescence. There is many other processes which are happening. So now leaves in plant. So the monitoring of vegetation by spectroscopic detection of electromagnetic radiation is powerful, non-contact and non-destructive method in the study of environment. And laser-induced fluorescence spectroscopy of terrestrial vegetation is an important aspect of active remote sensing, which provide a specific tool for assessing vegetation. So the growth of plants are based on the capture and transformation of sunlight by chlorophyll through the photosynthesis process. And the efficiency of this process depends upon the presence of sufficient amount of water, mineral, carbon dioxide, and light. So plant damage by pollutant, water stress, or pathogen usually result in a reduce of photosynthesis rate. So this is why the measurement of the depression of photosynthetic rate is an important criterion in the evaluation of accessory pigment to chlorophyll A and stress condition in plants. This is the chlorophyll absorption spectrum. And chlorophyll, it is a green pigment, as you know, which is found in almost all plant, algae, and cyanobacteria. And it absorbs light most strongly in the blue and red region. But it is poorly in the green portion of the electromagnetic spectrum. Hence, the green chlorophyll of the chlorophyll containing tissue like plants. This is the chlorophyll of photosynthesis. This is extremely important. In plant tissue, the light energy in the region of 400, 800 nanometer absorbed by photosynthetic pigment, chlorophyll A and chlorophyll B and carotenoid. It is used to drive the photosynthesis process, which provides chemical energy for plant growth. And the large part of the absorbed light energy is used for CO2 fixation. And photosynthesis takes place in the chloroplast, in which the photosynthetic pigments are localized. So this is here I explain a little bit the photosynthetic process. There is a photosystem 1 and photosystem 2, which are related respectively to the reaction center P17 and P680. We'll see after why these reaction center are important in the characterization of plant. No, in the process, there is a part of energy is lost during the migration from the pigment antenna to the reaction center. And it can be, I cannot see my slide correctly. And from the pigment antenna to the reaction center, and can be dissipated by a variety of photochemical process. And such process include the emission of heat and emission of 2 to 5% of the absorbable light energy as chlorophyll fluorescence. And the capacity of plant for photochemistry is limited and will depend upon a range of factors, including stress caused by environmental conditions. And if photosynthetic processes are very active, then red fluorescence will be low Conversely, if this process is inactive or implemented, then red fluorescence become important. So with this fact, chlorophyll fluorescence is used as a standard method for investigation plant class differentiation, chlorophyll content monitoring, and plant stresses detection, water deficit, temperature, nutrient efficiency, pollutant agent, attack from pathogen. Oh, what's the problem? So no, here we are going to show you the laser-induced fluorescence of leaf of some tropical plant at ambient temperature. Measured fluorescence spectra were then fitted with linear combination of Gaussian spectral function. For each Gaussian band, the fitting procedure set up the red and far red fluorescence spectra and provide the peak wavelength and the bandwidth and the amplitude of the band antenna.
Amadou, you have 2 minutes left.
Only 2 minutes. Okay,
yes.
So I have to go quickly. So I will skip a lot of part. This is the experiment with the fluorimeters that we have used. It is a very small. It is a compact fluorescence sensor which is based on violet laser. And here, this is the schematic of this fluorimeter. And in our system, we have we study the Moringa oleifera Andersonia digita. It is a tropical plant which we have collected in the garden in our university, in the botanical garden of our university. Here we have some of the results obtained for different kind of plant. And you see here when I talk about the fluorescence center, and we have to compare these two peaks. The comparison of this first peak and these two peaks give you the information about the state of the plant, the stress, the lack of nutrient, mineral, and so on, and everything. So I will go just to tell you for the conclusion of this part that the fluorescence is used for the specific identification of plant and also for the detection of plant stresses. So this is for the photodynamic therapy. We can use also the laser-induced fluorescence for photodynamic therapy. For that, we need a blue light here. This is a blue light, and we need a red light for the therapy. The blue light for diagnostic. For diagnostic, we have a curve here. If you have this shoulder, show the presence of a cancer, and you need a photosensitizer to. In our experiment, we use the ELA. It was in collaboration with the group in Sweden. And this is the experiment we have done in Senegal for the PDT. And now I will talk about the lips very, very quickly also, because my time is very short now. You know, and the lips, it is the principle of lips. It is to the absorption of photon, the terminalization of electron. Here you have our lips system. My students are working in it. It is a quick switch to 2 millijoule YAG laser at 1064 nanometers. And the LIPS experiment, this is the description of the LIPS experiment. We have a chamber for the safety. And here we have the application we have done in medicinal plants. Here also in Moringa oleifera, in Cumbertum, Kenkiliba, in Citronelle, and so on, we have a different spectrum. We want to monitor the presence of potassium. You know, potassium is extremely important, especially for medical application, for diabetes, and so on. This is the laboratory. This is also the application we have done in dental analysis. We have a collaboration with a medical faculty, and the girl is a student from Morocco who was working in my laboratory on the lip system. This is the spectrum of the cementum, the dentil, the enamel with a lips spectroscopy. Here we have some spectral some spectral fingerprint of nitrogen, of calcium, of carbon, and potassium based on the pathology which are in the teeth. So the last part is the multispectral spectroscopy. For that, we are in the network. We have done a first workshop in Ghana with a group of Sun Swanberg who have performed a multispectral microscope. This is the component of this multispectral microscope. So the application in the characterization of Plasmodium falciparum, which is the parasite of the malaria in a red blood cell. I can't show the. This is the experimental setup. This is my student, which was working in this system. This is the transmission mode and the scattering mode image of red blood spectrum infected by Plasmodium. No, red blood cell, I want to say. Here you have different experiments for that. And also we make a machine learning to make a differentiation, to know what is the quantity of Plasmodium falciparum in the red blood cell. For that, you need to make a machine learning. And this is the result of this machine learning. When we can see the red one are infected cell and the blue one are not infected cells. So you can make a differentiation immediately. We do it in a diffusion geometry, and the first one is in transmission geometry. Okay, and okay, this is all. There is some other plate, but I will skip that. So I just want to acknowledge now the Abdus Salam International Center for Theoretical Physics, the international program in physical science, and thanks to Sun Swanberg, Catherine Swanberg, to Johnny Mela, who was the head of the office, to Professor Denaro, and to all the African government. When we organize a workshop in a different university, we have a strong support of local government. I show you when the president of Senegal give a reception to the member of the African Physical Society. Okay, many thanks to our partners and sponsors. It is ICTP, Uppsala University, the LAM Network, the International Atomic Agency, and the University of Czech Anta Diop. Thank you very much for your attention.
Thank you very much, Amadou, for a very engaging talk. It was very nice to see a brief historical overview of the organizations you have been involved with, and also the range of very interesting research applications that you have shown us in malaria and also in photosynthesis. So especially the photosynthesis is very interesting for me, because you might know that I'm also working on photosynthesis. So we should definitely look into opportunities to collaborate. I will follow up with you on that. So let me first open the floor for questions from the audience. So we have time for one short question, or maybe two if there's a lot of interest.
Yes, good afternoon again.
Yes, Patrick, please go ahead.
Yes, but I appreciate the last speaker and the brief introduction to all international organizations that have been involved in one project or the other. And I want to also advise that there's a Nigerian Institute of Medical Physics in Nigeria. We have a president, I think he's working with the African Institute, or with African Institute too. So maybe those collaborations can be extended to this part of the country, where we can also hook on and see how we can develop this area of medical physics within Nigeria as we try to link up with the African bodies. Thank
you. Yes, I can answer.
Yes, you can go ahead.
Yes, thank you, sir. Of course, this is why I bring this presentation. It is how to to make collaboration with different African countries. As you say now, our friend from South Africa, you know, I give this presentation in one of the African Laser Center workshop. It is after that in South Africa they start to also to work on PDT, and I know people in I think it is at the University of Johannesburg, and also in Elizabeth Town, where there is people also working in PDT. So if in Nigeria you have a group there, it will be very, very important to work with you. And within the African Physical Society, this is one of the aim of the African Physical Society, to bring different research groups together, working together, and in organizing conference, in organizing common research program and so on. So yes, no problem.
Okay, thank you very much, Amadou. It was really a pleasure to have you here, and let's give him another round of applause. Thank you very much.
Thank you.
Okay, it's time to move on to our next speaker, and that is Ushi Patrick from the Cross River University of Technology in Calabar, Nigeria. And he's going to talk about the evaluation of temperature gradients within different head tissue layers exposed to radio frequency radiation emitted by GSM transceiver base station using the Penny's model of the bioheat equation. Quite a mouthful there. Right, so the floor is yours, Patrick. I see you are starting to share your screen. So you are welcome to start when you are ready.
I think Amadou, you have to stop sharing your screen.
Eh?
You have to stop sharing your screen.
Okay, because I cannot see
My screen now.
the Zoom. I have not the Zoom anymore.
Hold on.
Okay.
So Patrick, you can start sharing your screen.
Okay.
There you go.
Okay. Is my screen visible?
Yes.
Okay. Thank you very much for this opportunity. I'm Patrick from Nigeria, Cross River University of Technology in Calabar, Cross River State. Well, a member of the Nigerian Institute of Medical Physics. We hold our conferences yearly around November. Because last year we couldn't because of the pandemic. So I know that this year we are planning for another evaluation of the temperature gradients within different tissue layers exposed to radio frequency radiation into the adjacent transfer station. My presentation will be more of mathematical. It's an ongoing project that we have concluded, but we are about testing. So when this conference came up, I said I should share with other.
Sorry, Patrick, could you move a bit closer to your microphone? I can't hear you very well.
Okay. Can you hear me now?
And that's better, yes.
Okay. So I said the project is ongoing. We have done some evaluation, done some analysis, some simulations, and we have arrived at a result. We are in the testing process. So as for now, we have tested with the GSM. We are thinking of going to the hospitals and do some other tests. And the project is more of mathematical, so I will show you some mathematical equations and the simulations we have done. Well, in the last decade, we know the importance of cell phones and the advantages and how the rush, how the increase has gone across countries and continents. So it's advantage. But with that, we have not really sat down to look at the disadvantages of those cell phones we use at every point in time. You know, definitely they are. Cell phones are transceivers, they are packaged with both receivers and transmitter antennas. And even the cell towers too. They also have their own different level of transmitting frequencies and receiving frequencies. Every country has its own bandwidth. Like Nigeria, our anchor organization is the Nigeria Telecommunication Frequency. spectrum allocated by NCC. So for every network we have within the country, have their own bandwidth for the GSM 900, GSM 1800 megahertz, and the current one, which are expanding up to 2100 megahertz. They have a different bandwidth regulated by NCC. We have the Glo network, the network, the Airtel network, and the Planet network. The other minor networks, these ones are the major ones for now in the country. So we looked at other standards outside here, and we said, okay, we should aim at taking measurement of radio frequency radiation across field within these transceiver stations using the transceiver network, which is the cell phone too, because they are both transceivers. They receive and transmit radio frequency radiation across field. But we try to limit ourselves to 100 meters for now. If we have to progress in the project, we will increase our length of measurement. We're also looking at to investigate the exposure level of human tissue around the GSM transceiver base stations in the Fresnel region. The Fresnel region meaning that they are very close to the transceivers, the other antenna of the cell phone, the antenna of the base stations. Because you know, the cell phones without keys or without earpiece, they are always very close to us within 2-3 meters. So every other biological tissue or body tissues, in case may be. To evaluate the level of exposure of human tissue population within the South, we use the South region because this is our range. This is where we have to cover now. As the project goes on, we are thinking of covering even outside the country within the scope. This is the South region. This is the map of South, and these are the areas. Those we are seeing the areas and locations we decide to do our measurement. So what do we use in our measurement? We use electro smoke that covers a range of up to 8 gigahertz of radio frequency. The second display here is how it's displayed when it synchronizes those radiation from the field. This is the display mode that we see. This is a software that has been used by international regulatory commissions to synchronize this. So they simulated this work we have done. They've already done in simulation. But we want to do ours in the field, which is more practical. We call it in situ. To compare with all these simulations to see if these simulations are really obtainable and we can really apply them. Then we have gadgets that we use. It's just the few method. We have SAR for every cell phone, every transceiver gadget has a specific absorption rate. So for every cell phone, for every transceiver base station, they have their specific absorption rate. So we went to the field, we took readings to see how we can evaluate that SAR in the field and use it for our simulation. Our interest is in the thermoregulatory system, the human thermoregulatory system, temperature gradient of the human system. Because the human system is divided into layers. The commission look at those effects holistically, the body system, how does it react to it? But we discover that layers of the body system react to this radiation differently because of their mass, their density, their specific capacity. And so we look at those small details to see how the layers are affected differently. So we started with the energy balance equation, which is the equation we are saying, to the psi equation related to thermoregulatory system in equation 2, equation 3, sorry. Then if you evaluate it, applying necessary boundary conditions, you want to solve this equation 3, you arrive at equation 4 using the necessary boundary conditions. This one's we've done that, but we might have the time to do those calculations here. Then our interest will now be this equation we call the pin-bar equation, pin-bar equation. So we call it PBHE, which is an equation from here, 1000. That's material. You see this as the pin-bar equation, where TB is the temperature of the blood, H is the metabolic generation or the heat source, then B is the system. Okay. called thermal conductivity. Like Fourier law for heat convection, which is this that equation was seen in equation 6. But you know that this electromagnetic radiation emits from the source, expanding phone or station. Since the EM emission was observed in the fixed position, it does not vary with time. At every time, it flows at a steady rate. So if steady flow is applied here, meaning that this change with respect to time will be equal to 0. If you apply this to that inbound equation, you discover that if you evaluate all that equation properly using all the necessary boundary equation, you discover that we arrive at equation 7, temperature gradients, or temperature difference, or temperature flow, because the equation where rho is defined the density, the volume size, the source, and the volume flow, the flow rates within the system, and Ts and Tb are different temperature of the system, the body system, and surface of the biological system. We also look at this equation 8. This equation 8 almost did the same thing of what we've done, but is we evaluated the pinball equation that was used in this work to analyze the temperature distribution of human tissue. Equation 8 is a Fourier series. The difference between what we did and what we have in equation 8 by Manuel et al. In 2013. We use Fourier series solution to solve using finite element, which is more of a computational method. What is the computational method? Arrive at a solution and try to test it. So we also did that and compare us with its own to see the correlation, see how that is. We went back to the basics. The basics are the different tissue layers within the body system or the head system, where most of the time the phone is always close to. You have air between the head and where the phone is. Air space. You have the skin of the head, the skull bone, the dura mater within the head, the cerebral fluid, which is inside that before the brain. The brain is the last substance or last tissue in the human head. So we do some simulation there in the field situation to analyze different thickness of the different tissues before you get to the brain of the human head, where most of the time we find bones very close to the particular emission of distribution towards the head as you receive this course. If you look at this diagram, you see the different layers with their distances, different distances from the surface. The surface is, look at this one shows the phone, this one is an airspace. It can be 2 meters, can be 2 centimeters, can be depending on where the phone lies. But we are using the near field cables with the Fresnel case at 2 centimeters maximum from these points to the head. So you can see the different tissues as you get to the head. We arrive at this. Operator 1 is one of our networks here in Nigeria. I've displayed 4 different networks, but I didn't want to call names because of the ethical condition. So operator 1 is one of the network, operator 2 is one of the network, operator 3, operator 4 is a different network. We collected those readings and used equation 7 as our heat source. In rho SRA, which is SAR, is a heat source. It's a point of contact between that emission and the body system. So we use operator 1, we collected those readings and calculated the temperature variation, temperature gradient, the temperature difference as it's across the different tissue layers. So we discovered that in air, if you calculate using the free space, which is air, we got this in skin, in fat, like that. So the second operator is there, third operator is there, fourth operator is there. We now compared this work we have done with Emmanuel et al. That use the Fourier series, the computational method, to arrive at the result. So we arrive at this. For operator 1, we discovered that operator 1, operator 2, operator 3, operator 4, and our result was in synchrony. Our estimated result was in synchrony. We find it all lying within one plane. Though the there's this uniformity between the man on the tour work and what we did from the field and using our own model equation from a pin bias equation and applying joining with the Fourier series equation. We discover that all those operators, all those parameters lie within this line. And but for Emmanuel de Tor, it has the same trend, but the values are different. Well, if we look at this, we discover that in the field, the field result will give us more accurate value than the simulated result. So we are seeing that the difference because of that simulation, since he didn't use the different tissue layers, he just generalized here and say, okay, this is how the trend will be. But we now pick it individually and say the skin is different, the head is different, the fat bone is different, the cell is different, and the fluids, as in just the brain, they're all different. And that's why we'll come out with this result. You can see that the synchrony between what we got from the field and what we estimated using the organization simulated result. This is estimated using the estimated tool used by National Regulatory Commission. So, meaning that our Bowles equation that we derived is consonant with what the manual taught did and what the commission is also using. So in our result, so the temperature gradient is maximum in the brain and least in the renal fluid. That shows that the blood flow rate must high depends on radio frequency radiation. Meaning that where there's more blood flow, there's more effects of radio frequency radiation from any. To show that the most variable tissue when exposed to radio frequency is the brain, not other tissues. So we're specifically dealing with the brain, which is when it's more affected, so the brain should be more protected. Thus, the modified Bowles equation accurately explain the exposure to radio frequency. The near field. This is near field now. As the project continues, we are trying to look at the far field and see the correlation between the two results. But for now, the near field, there is no correlation using the Bowles equation. The group of agreement for that model estimator. So we're also looking at that if we use this equation 7 as a heat source, it can also be applied in hospitals because we have already tested it now for radio frequency radiation. We can also use this component as our heat source, so that before we apply any radio, any diagnostic using X-ray or any photo, we can also calculate using that same heat source before it is administered, so that it will reduce the effect of burns, the effect of the effect on cells on the body tissue before any radiological or any diagnostic can take place. So what we are achieved here is that this modified Bowles equation in our own way can put in an heat source here can also reduce the danger of the effects of already any diagnostic equipment on the biological tissue or human tissue. So,
Patrick, you have 2 minutes left.
All right. So as we are moving on, I know that we are concluding with testing in the hospital, but for now, we have only used this. Thank you very much.
Hey, thank you very much for wrapping your talk up so quickly. And the floor is now open for questions.
So it seems like these days, cell phones do not depend upon you holding the phone to your head. And actually, I think they don't work as well when you hold it up to your ear. So does that make them qualitatively safer? Because it seems like the penetration of heat into the brain was much more than any other tissue.
Well, the advice. Oh, can I respond?
Yeah.
And the advice is that the phone should not be so close to the head. But you know that all the regulatory agencies look at it holistically and say it affects the head, it affects the tissue, but they don't specify to what extent does it affect the first tissue, the second tissue, and the third tissue before it gets to the brain. And that's what we are trying to look at. But it's advisable that this cell phone should always be out. There should be space, there should be airspace between the cell phone and at least 5 centimeters away. Okay.
Yeah, I see that our next speaker is not here. I did see him a while ago, so that means let's continue with the questions. Yeah, so the floor is still open for questions.
Yeah, where did he go? He was here. I gave him the.
Yeah, I also saw him. Maybe he's experiencing some connection problems.
That sucks. Huh.
Yeah, and then the very last speaker told us that he would be around after 4:45. Yeah, so that is Malik Maza. So I think what we could do in the meantime, Lawrence, you also said that there's a lady who wanted to talk for a couple of minutes. We could give her.
Whitney, are you still here? She's. Whitney, are you here? I see your box, but let me.
And what we could also do
She might have stepped away.
Now, while the president is around, why don't we open the floor and encourage the participants to ask some questions? Now, let's first hear whether there are any more questions to Patrick. If not, Patrick, thank you very much for your talk, for a very, very interesting talk. And let's give him another round of applause. Thank you very much.
I think I saw Whitney just come back.
Yeah, I saw the same.
Oh, there she is.
Hello?
Yeah.
Yes, so sorry. I'm having some really strange connectivity issues and my computer is having some issues. But hello, everyone. My name is Whitney Stevens Sostre. I'm a PhD student at the University of Wisconsin-Madison. A wonderful talk today. I will admit a lot of it went over my head. My current training is in electrophysiology. I study voltage-gated potassium channels. But today I came here to speak to you about the Black in Biophysics movement that was we recently founded. So I am the founder of Black in Biophysics, and we are hosting and organizing a Black in Biophysics week from May 10th to May 16th. We launched our organization at the Biophysical Society meeting this past month. And currently we have our first organized activity was a happy hour for Black biophysicists and others who are also allies. That was hosted at a virtual platform called GatherTown. And so here are other organizers that we've been collaborating with. All of us, we're at the United States, but we are from different institutions, including the University of Washington, University of Michigan, and Torres Velo, he manages grants at the NIH, also Northwestern University. And so this has been kind of a small 10 people group that we're kind of working really hard to make this happen. And so I wanted to share what was the proposed agenda for the Black in Biophysics Week. And so you may be familiar with Black in Neuroscience or Black in Neuro, which was one of the first larger disciplines that organized to make a Black in week. And so because this all started kind of around COVID time, it is more of a social media or virtual week. And so that's kind of what is reflected here as we've organized. And so for the first day of the week on Monday, we typically, and other Black in X groups have done a roll call. So this means that we'd be highlighting Black biophysicists at all stages, whether it be undergrad, graduate, scientists, academic researchers, whatever it may be. And this is meant to for us to have more exposure. If many of you have attended the Biophysical Society meeting, there is a lack of diversity and representation. And so this is meant for all of us to find each other within our community. And so one of the ways we'll do that is to take over the Real Scientists Twitter account, which has thousands of followers, and continue to retweet the different profiles of different Black biophysicists around the world. For the second day, we have kind of a next generation panel where we want to organize a panel with graduate students and postdocs, and via Zoom have a panel where we would share our personal trajectories, what got us into science, what got us into biophysics, with younger generations, specifically targeted towards middle school and high school students. To achieve this, we are contacting different schools because it is like an within the week day. It's a Tuesday, so we are hoping to collaborate with teachers that want to offer their classroom time to share this panel and have students ask questions. For Wednesday, we have a panel with different scientists that have taken different career paths from what is the normal or expected trajectory, which is academia. And so these scientists would be in academia, but also different roles in industry, in science communication, and in patents and grants. Specifically from the NIH, we're hoping to have a speaker join us. We're also hoping to have, again, like a diverse panel in each of these different panels that I've described. On Thursday, we're planning share science. So we're planning to do a social media competition, scientists sharing their biophysics research. But kind of right now, the idea is that we try to explain in layman's terms, so it is accessible to people who are not in science. And so it's kind of more of a science communication planned day. There will be judges and also prizes that will be sent to the best presentations. We're also hoping to organize on this Share Science Day a series of talks that are broad in disciplines within biophysics. On Friday, since it's the last day of the week, we have a networking event. We're proposing to do a Gather Town happy hour again, where you would meet in a virtual space and have an avatar to walk around, interact, meet people who are doing biophysics, hopefully with the idea of expanding collaborations between different labs. For the weekend, we have planned physical and mental health activities. So for Saturday, we want to host a virtual Zumba class or something related to having this work-life balance between science and health. And finally, on Sunday, we'd like to focus on mental health. So we'd like to collaborate with the Black in Mental Health organization for them to host a series of of talks related to mental health and the importance of maintaining it throughout your career. And we're also hoping to host a virtual yoga session. And so that's all that we have proposed. Moving forward, we are still looking for speakers in different fields or within biophysics, so they can share their career trajectories and research. This is specifically for the next generation panel, and also for the Share Science Day. We're also currently looking for sponsors to fund our Black in Biophysics Week, with hopes of being able to pay our panelists, our participants, for their time and their work during our week. And we're also looking for early career organizers to help us plan and execute this week with success, specifically looking towards graduate students and postdocs who would like to contribute in any way. And that's all I have for today. I just wanted to share briefly what we were planning and open the floor if anyone has any questions or suggestions. Happy to answer them here or via email. We've primarily been using our Twitter account as the means of communicating with our community, but you can also email us at the email address shared here.
All right, thank you very much, Whitney. So as you said, the floor is open for questions.
And I will add, please do share broadly within your communities. A lot of us are here in the United States, so I think we're really trying to expand and share that this is happening and that we want our community to participate and engage. So please do share that this is happening in the week of May 10th to May 16th.
This week is happening in May?
Yes,
but in
in
your
May.
slide, I see March.
What? I'm sorry?
In your slide, it is written March.
Oh, I am so sorry.
Not
It's in
May.
May.
You see, everywhere.
Oh my goodness, yes, you are correct. I'm so sorry. It's
okay.
May.
It's
Sorry.
May. Okay, thank you.
I don't know how that happened. But yeah, sorry, it's up here, May 10th to the 16th. And this is the main slide that we've been sharing with our network to announce the week in May. Thanks for catching that.
Okay, thank you.
And we have to wait a few minutes for the next speaker, so we have plenty of time to ask questions to Whitney. So please do use this opportunity.
Who is the next speaker?
That's Malik Maza.
But it's
So he has a meeting up till 1:45.
It's not written in the program that I have.
Yeah, he's a late addition. And then there's one speaker who, I guess, dropped out.
Yeah, he was here earlier.
He was here, yeah. I was like,
he disappeared.
I just sent him an email to see if he is coming back. But maybe he suffered maybe power outage or something. Whitney, let me ask you, do you know do you have of your team, what is the distribution in terms of fields of quantum biologists, structural biologists, molecular, cellular? Yeah.
Yes. So most of us are, I think, more in the molecular biology. Dr. Davis, I think, is closer to structural biology. We have mostly electrophysiologists, I will say, because as I initiated this movement, that was the network that was available to me. And so people who reached out and wanted to contribute are mostly electrophysiologists. But yes, I would love to have representatives of each of the different fields that are, biophysics encompasses. So yes, if there's anyone that you would like to share this opportunity with, I would love for our organizers in our board to also be more representative of biophysics as a field.
Right. So I imagine in the United States, there's the same problems as everywhere. How do you get students in traditional fields like chemistry and physics, I guess biology, to come to biophysics?
Yes, I completely agree. That's part of one of the things that we were thinking of when we planned for the next generation day is to introduce biophysics. I feel like a lot of scientists, myself included, I'm a biologist, and I'm doing my PhD in neuroscience. But I am now in biophysics because of the nature of my work with ion channels. But I did not know of the field in itself until I joined my PhD lab. So I think we do have the same challenge of exposing chemistry students or even molecular biology students to this field, and how a lot of what is being done in research is actually biophysics as well. But yes.
All right.
Right.
Any other questions?
I'm sorry?
They're just asking if there are any other questions.
If there are any what? Elections?
Questions.
Questions.
Oh, questions. Sorry. I can't hear. But if not, I will stop sharing my screen, and please do reach out to me or to the group via Twitter or via our email.
Let me just make one comment. It's just reminded me of electrophysiology. I just I noticed yesterday that Shaka Fatah is back. Who was, I guess, started the Brain Initiative in the United States. Yeah, I guess he went
retiring.
He had to go away for a while, and now he's back. So one sort of, I guess, overriding application of biophysics is in health and disease, human and animal health, and to some extent plant health. What are some unique perspectives that Black biophysicists can bring to those things, both in the United States and in Africa?
Right. I think, just, I guess, answering very broadly, I think that for one, in science in the United States, and I'm not sure if it's the same in Africa, I haven't visited the continent, there is a lack of representation of Black scientists in general, in every field. And so I think as biophysics in general, I think we do have a lot of contributions in different fields that are more related to biology, physiology, and the pathology of disease. And I think one of the things we can do is start developing more grants that are biophysics focused, but targeted to different disease-related or health-related problems. I'm not really sure that how well-funded biophysics is as a field compared to neuroscience, for example, that has this whole brain initiative that funds neuroscience research.
Right.
So yeah.
Okay. I still do not see Fernando. I sent him an email. He did not respond. He hasn't responded yet. So maybe he just evaporated somewhere.
Yeah, so that leaves more time to ask anything to Whitney. So if anyone has a question, please do use this opportunity.
I'm sorry that we didn't get enough time to give our talk, and now we have plenty of time.
Yes, that's because the second last speaker dropped out, and the last one is a bit late.
Yeah.
Pity,
Right.
Yeah, well, Amadou, we could give you 5 more minutes while we're waiting for the last speaker. Why don't we do that? So before we do that, so Whitney, it was really nice to have you here.
I just say that, but it's fine, no problem. It's fine.
Yeah, well, I think we could make very good use of this time to ask the president of the African Physical Society any questions while you're around. It's really a privilege to have you here. So I think let's open the floor and ask the president
No, it's
anything you would
okay.
like to know.
No, I have
Or if anyone has a question to Whitney, please take this time.
I have a question to Whitney.
Okay, yes, please go ahead.
Your meeting is a virtual meeting or not?
Yes, it's entirely virtual.
And one have to register for that.
Yes, that's the idea. Since we do have some time, we're still working out the logistics of how to register, but we recently made our website. And so we will be sharing in Twitter the registration link and further information about the proposed agenda. The agenda I just showed to you all today is not public yet. So we were hoping to gather input from the community here. But yes, it's virtual.
You are affiliated to some physical organization or to medical organization?
To neither. So we aren't, to clarify, we have, by the Biophysical Society has been helping us with, I guess, sharing our group, but we aren't technically affiliated to any institution. It's more of a movement or organization to recognize and highlight different scientists, Black biophysicists within the field. So we don't have an affiliation per se. So everyone and anyone who wants to contribute to the cause is welcome.
Okay.
Thank you.
Okay, I see that Fernando has joined us again. So yeah, let's invite him to share his screen. And Fernando, if you can hear me, you can share your screen and then start. I would like to thank Whitney very much. It was really nice to have you here. It was a very nice addition to the program. So thank you very much for your contributions. Right, Fernando, we've been waiting for you, but the floor is now open for you, so you can share your screen and start your presentation. Right, so this is Fernando Mukomole from the Eduardo Mondlane University in Mozambique. And he's going to talk about the efficiency of obtaining chicken chicks from a dimensioned automatic biophotovoltaic incubator. Right, I can see your screen. I can actually see myself. So I think you just have to switch over to another window so that we can see your presentation, Fernando. Fernando, please also unmute yourself, then we can hear you. Okay, so at the moment I think this is your Zoom screen. I can only see a picture of myself. So try to switch to another window where we can see your presentation.
I think you have to click on the actual window of your presentation. So the PowerPoint window, you have to click on that.
So Fernando, the easiest might be if you stop screen sharing and then start screen sharing and then click on the window where your presentation is.
There we go.
Yes, there we go.
Yep.
Right, so you can switch to full screen mode. Okay, there we go. So the floor is yours.
Check your microphone. Make sure you're not muted.
Yeah, he's not muted, but maybe you have some audio problems. Now, Fernando, we can't hear anything. You're not muted, so I think your audio is not working. If you go to Zoom, go to the microphone and let me see, I think you go to test speaker and microphone. Oh no, go to audio settings and make sure that you've selected the correct microphone. So Fernando, we still can't hear you. Have you been able to test your microphone? So if you go to Zoom, click on the microphone button, or on the arrow on the right of it, and click audio settings, and there you can select the right microphone. We actually don't even have to go to audio settings. So if you just click on the right of the microphone, there you can select a microphone.
Is that his background that we're hearing?
Oh, that is his first slide. So Fernando, you're now muted.
So I muted him. Now I unmute him. So we can hear the background of where he's at. We just can't hear his voice. It's really strange.
And I also see that Malik Maza also hasn't showed up yet. So let's try a few more minutes with Fernando.
Okay.
So please do try to sort out your audio, and then hopefully Malik will show up in the meantime.
Or at least he can just scroll through his slides and we can read them. Because it seems like an interesting talk.
Yeah, that's a good suggestion, Lawrence. So Fernando, if you do not, or if you aren't able to sort out the audio, please give us some time to read through your slides, and then we could ask you some questions in the chat at the end.
Is he still here? Yep, still here. So Fernando, if you go to the full screen, your presentation, and just allow us to read your slides, just read them. We're having a problem with the audio, but we can see your slides perfectly fine.
Okay, Fernando, you're unmuted now. Try to speak and we will tell you if we can hear anything. No, we still don't hear anything. Okay, I think let's do what Lauren suggested. So go to full screen mode and slide through your presentation. So I think most everyone should be done with slide 2 now. So you can then start with slide 3. Give us a bit of time to read through it and then go to the next slide. So Fernanda, could you go to slide number 3? Fernando, can you actually hear us? If you can hear us, please let us know in the chat. Okay, Lawrence, I don't think Fernando can hear us.
Okay.
He's not
Yeah,
responding to our requests.
he must
And
have
Malik is also, he hasn't showed up. So shall we then close the meeting here? Or close the conference?
I guess so, yes. Yeah, well, let's do that, and we can have Malik at us just a regular Zoominar in a few weeks.
Yes, definitely.
Okay.
Okay, yeah, so let me formally close the conference. So we have now come to the end of the first Biophysics in Africa conference. And I would like to thank everyone for their participation to make this conference a great success. We had an amazing program of talks by both African and some very prominent non-African speakers, covering a very broad range of topics within the broad field of biophysics. And we had a lot of excellent talks. For me, this conference was really very enjoyable, and it has also displayed some of the growing research excellence on the African continent, about which I'm also very glad. I was very glad to witness all of that. So yeah, I would like to encourage you to complete the conference survey, which we sent to you yesterday. And from the responses we already received, it was clear that this biophysics conference has to be made an annual event. So I hope to see everyone back next year again. And in the meantime, as Lawrence has mentioned, we are going to host some Zoominars and hopefully also get some courses being published on our website, which you can also follow. So yeah, we're going to develop different ways to advance biophysics in Africa. And if you have any suggestions, please do let us know, because we really want to grow this community, grow the awareness of biophysics on the continent, and hopefully soon we can also have some strong political support and some funding and all of that. Right, so with this, I would like to close the conference. Thank you very much, everyone, for joining. And yeah, we hope to see you back soon again. Any last word from you, Lawrence?
I think Amadou wanted to do a last word.
Okay, yes, please, Amadou.
Amadou, are you waving, or did you have something you wanted to say? You're muted.
I'm saying goodbye.
Okay. All right. Goodbye, everyone.
Okay.
All right. Cheers.
Goodbye, everyone.